Good afternoon and good morning to everybody watching us. I give you a warm welcome to another one of our live interviews, the live interview series by the ON Foundation. Today is my big pleasure to talk to another friend of mine and to another pioneer in tissue regeneration, and honestly one of the leading experts in ortho regeneration in the world. Professor Ivan Martin. He's director of the Tissue Engineering Research Group at the University in Basel. He originally studied biomedical engineering in Genoa, and from there he moved on to the United States, where he spent 3 years at MIT and Harvard. Back in Switzerland, he became appointed as professor for tissue engineering, and he also became chair of the European section of the TERMES Society, the Tissue Engineering Regenerative Medicine Society, where he now coordinates strategic alliance committee. He is highly productive in research. So he's an author of more than 250 peer-reviewed papers. He has an incredible age index of 83, probably increased in the meantime. And he's a real inventor with his name on more than 10 patent applications. As an inventor, he's used to think out of the box. And as we will see today, he's also thinking out of the knee. Today, we will talk about his fascinating on-stage talk that he's given last year with the title, How the Nose Could Regenerate the Knee. But before we start, I'd like to encourage everybody out there following us on Facebook and YouTube live. Please, maybe in the beginning, introduce yourself, tell us where you are, who you are, and then start questions as soon as they pop up. But now let's start. Warm welcome, Ivan.
Thank you, Kai. It's a nice opportunity to interact with people these days. So thanks for organizing and taking the initiative for it.
Thank you so much for taking the time and taking us home.
In the real sense, yes.
Yeah. Before we jump to exciting science, I'd like to ask you 2 personal questions, if you don't mind.
Sure, I'm not prepared for those, but...
It's not intimate, it's just personal. So I'm always curious what made people entering the career. What made you starting or becoming a biomedical engineer?
It's actually an interesting question also for myself, because I studied electrical engineering in my undergraduate studies. And I was doing my master thesis dissertation on calibration of robotic systems for space applications, so something which had nothing to do with skeletal or biological regeneration. But it was in investigating from an engineering perspective how neural networks function and can be programmed in artificial intelligence that I recognized in my soul an interest in more the biological control of these networks as opposed to a informatic computer-based control. And there I got fascinated by the interaction between cells and how they send neurotransmitters, and got fascinated in biology and entered this field. Contingency brought me in the field of regeneration and osteoregeneration, but this is only a detail.
Oh, thank you. It's always nice to understand the motivations. Yeah. Maybe another one, and that's already related to the talk you've given at ONstage. Have you already done yoga today?
Not really. I typically reserve a physical exercise session in the late afternoon, so it will come after this
okay, good,
interview.
Good. So we try to be in time for everybody so that you can go for sports. Okay, now let's come to this interview and the scientific content. There are some viewers out there which might have missed your on-stage presentation. Can you maybe within 1-2 sentences explain your idea, the idea behind it?
So we know that cartilage and articular cartilage in particular does not have an intrinsic capacity to regenerate. And among all the possible strategies that are being investigated and which are possibly valid, our approach is to introduce into the joint a component, so some cells that have the capacity to bring the signals and to instruct the resident cells to regenerate cartilage. And this is not a new concept. Many other groups have tried different approaches based on living cells. Our distinct strategy is to take the cells out of the cartilaginous part of the nasal septum, because we have recognized that since these come from the neural crest compartment that develops primordially and with a hierarchically superior biological properties, these cells could have a strongest induction potential for regeneration. This was an initially quite brave and let's say adventurous approach, but we recognized in several different models, in vitro, in vivo in animals, and ultimately in vivo in patients, that this is actually correct, that the cartilage cells from the nose have some special signals that can induce regeneration, and that's why we are testing them to repair articular cartilage.
Great, thank you. So I'm sure we come back into these details during our interview. But before, let's go one step backwards. And I think there's several techniques out there where people try to repair cartilage defects or regenerate. What is missing there?
So to be honest and fundamental, I think what is missing is a clear understanding of what is missing. So we still have not understood why cartilage cannot regenerate, in contrast to other types of tissues, skin, bone, liver. Is it because there is a cell type which can be attributed having stem properties which is missing? Or is it because of the environmental conditions that do not support regeneration? What is the role of inflammation that occurs in an injured joint? Is this the type of environment preventing a regenerative capacity of the tissue? So until we truly understand this and we can molecularly control the pathways which could lead to regeneration, I think all the attempts to regenerate cartilage will remain a trial and error approach. I'm very skeptic in this sense, but this is how it is. It does not mean that there cannot be valid solutions as of today, but these will never be optimal solutions. They will never be broadly applicable with certainty of success until we understand what is missing and how to molecularly control what is missing.
What can be achieved as of now? If I fall off my bike today and I get to the clinic, what can be done?
So as of today, we can fundamentally distinguish between two different types of bad events for cartilage. One which is related to injuries, so to traumatic events. You fall out of your bike, you have an injury at the articular cartilage. It's a fresh focal lesion. And then you have instead some signs of continued chronic degeneration, inflammation that leads to osteoarthritis or similar types of pathologies that involve a broader compartment, less focalized, and clearly not fresh. For the fresh injuries, depending on the size of a lesion, you may well be satisfied with the current procedures, even with marrow stimulation techniques, if these lesions are rather small. But when the lesions tend to get larger and when they are accompanied with other types of lesions like a meniscus injury or a ligament rupture that lead to instability of the joint, then it starts not to be untreatable, but to become unpredictable. What will be the outcome of the treatment? And we see here even less young people recovering very well, and in contrast, some young individuals treated that do not manage to reconstitute their cartilage. So this is the area where we need to find more efficient solutions. And then if we go into the stage of more chronic degeneration, then actually most of the times what people are trying to achieve is to find ways to delay the ultimate solution that will be, unfortunately, a prosthetic joint replacement.
That takes us back to what you said, the lack of understanding what's actually happening. And that's also a question from the audience. And she's asking, what would be an appropriate model to work on this underlying question, what is missing?
It's probably the question to be asked. So how can we learn this fundamental knowledge? And many animal models that have been popularized in the past and have been advocated to approach the biological and mechanical properties of human joints are more and more being recognized as non-suitable. So animal models, even large-size animal models, in general, in my view, should give proofs of feasibility of the concept of safety, but they can hardly be predictive of efficacy. In vitro models may sometimes be more informative in this respect, because at least we can use human-derived cells, and the biology and the mechanisms of biological control of human cells are dramatically different from those of animals. But obviously, in an in vitro system, you miss several components of the environment, of the mechanics. There are some interesting new even organ-on-chip models, including mechanical loading systems that try to approach and simulate the biology of the joint, but obviously we are still quite distant. So ultimately, the real model to play with is ourselves, is the patients. What we have to do, however, is to be extremely careful and to implement strategies of therapy in critically designed clinical studies, which are sufficiently powered with appropriate controls, prospective, randomized. And so I would advocate that we have to be more systematic in the way that we treat patients to derive as much information as possible and to learn. And there are new tools nowadays that technology helps us with. We have, for example, the possibility, even out of a few microliters of synovial fluid, to screen for the presence of up to 10,000 cytokines. And if we managed to do and introduce these analyses in the regular treatment of patients, we can learn so much about correlations between the status of the joint and the outcome after a certain treatment in clearly defined patient categories. Because the other obvious and I would say normal mistake that we tend to do is to group all cartilage repair patients in one box, and this is clearly not the case.
That's a great topic, and maybe one of its own, which we should discuss in the next interview of a series. But let's come back to your project, the nose to knee. Can you quickly describe what you're actually doing?
So in this project, I can refer actually to a clinical study which is ongoing and which is funded by the European Union, and that targets the treatment of a total of 108 patients in 4 different countries, in 5 different clinical centers. And these patients have all focal injuries, so confined lesions of the articular cartilage in the knee. From these patients, we take a small biopsy from the cartilage nasal septum. It's a 6 millimeter punch biopsy. Out of this very small piece of cartilage, we isolate and expand the cells, the cartilage cells, and we reach from a couple of thousand cells, we reach up to tens of millions of cells. So we have a sufficient number of cells to colonize a support material, a scaffold, so-called, and the density of the cells, this material, is high enough that it supports the development of a new tissue. So we ultimately grow a patch of cartilage starting from the patient's own cells. This process in total requires 4 weeks, and obviously all the methods and procedures are carried out in clean room facilities under strict control of the quality management system that is approved by the regulatory authorities. And the resulting patch is then implanted into the articular lesion of the patient, shaped intraoperatively to match the focal the size and shape of the focal defect. And this is done in a mini arthrotomy. And then the patient is exposed to a very clear and precisely defined regime of postoperative rehabilitation, which is very crucial in allowing the graft not to delaminate and to be lost in the joint, and at the same time to be stimulated by the mechanical loading of the own body weight.
The patch, I might have missed it in your explanations. Is it just a cellular patch, or do you use a scaffold?
So we use a scaffold as an initial template that provides the size and shape of the patch, which is, by the way, a 3 by 4 centimeter rectangular piece. And the cells are seated on top. The cells produce their own cartilage matrix, and then slowly the scaffold, which is made of a collagen material, resorbs, degrades. So there is still traces of this scaffold upon implantation, but over time, the scaffold itself resorbs, and only the products of the nasal chondrocytes, of the nasal cartilage cells, remain in the joint.
There's a question from the audience, from Fatime, and she asks what kind of scaffold you use. You mentioned it's collagen. Are you producing it yourself?
So this is a commercially available product by the company Geistlich in Switzerland. This is a membrane which is made of type 1, type 3 collagens. It's a bilayer scaffold. So this is an interesting detail that so the membrane is not uniform. So there is a layer which is more open in the accessibility, in the porous space, and then there is another layer which is less permeable to cells. And so we obviously bring the cells only in the permeable part of the scaffold, so that the cells can colonize the scaffold and build a cartilage tissue. However, this is well connected with the underlying part, which is less open in porosity and can support an easier fixing of a graft into the joint.
And how about orientation? I think if you've got a layer, you can put it this way or that way. Are the cells showing to the interarticular space or to the bone?
So the cells look into the bone, and so they directly interface with the surrounding cartilage tissue or underlying subchondral bone.
Okay, thanks. We talk here, we're using nasal chondrocytes. What similar approaches have been done with articular chondrocytes and with the ACT technique. What's the difference?
So obviously, before we started working with nasal chondrocytes, we have done years of studies using articular cartilage cells. And these studies were accompanied by a strong frustration, because we were always striving to generate biomechanically functional pieces of cartilage, so patches of cartilage. And we were trying to develop protocols to convince cartilage cells from the joint to generate these types of tissues. And once we were successful using cells out of one patient to generate a nice piece of cartilage, the same protocol would not be successful using cells of a different patient. So there is a strong inter-individual variability in the performance of these cells, which is clearly linked to the age of the individual, but beyond that, even cells from quite young individuals have unpredictable variability in the behavior. And so we could not manage to generate protocols, convincing protocols, to reproducibly generate the same quality of patches of cartilage that we generate using nasal chondrocytes. On top of the performance of these cells in vitro, so to generate these pieces of cartilage, what we also demonstrated in animal studies is that the persistence and the quality of the repair that is generated from nasal chondrocyte engineered cartilage was superior than the one of articular cartilage derived grafts, even when we were successful in generating them. So we had good arguments to convince the regulatory authorities to try something which was first in human, so to put some components of the nose into the knee.
There's an interesting question coming from Kiki Häusler. He or she really appreciates the approach and the mentioning of the importance of the mechanical stimulus to joint, and especially the cartilage. Do you also stimulate the scaffold beforehand, before you seed it to the cartilage?
Yeah, so this is also another very critical point. So first of all, and this was one of the main arguments before we could introduce this procedure in the clinic, was will the nasal cartilage cells respond to loading in a physiological way as articular cartilage cells? Because clearly, unless we touch too much our nose, these are cells which are not exposed to loading physiologically. And the answer to this question was generated using very sophisticated models in vitro. So we were exposing to compressive loading, to shear forces, the tissues that were generated out of nasal cartilage cells. And we recognized that the tissues were reacting to these mechanical forces similar to how articular cartilage cells react, including in the production of lubricant proteins, which are not present in the nose and which are pivotal for the functioning of the joint. So somehow there is a common regulatory mechanism also to mechanical loading of the nasal cartilage cells. After demonstrating that they have the capacity for response to loading, we had to design still a strategy to implant these grafts in the joint and decide when to expose them to loading. So from a practical standpoint, starting to stimulate these tissues during their development in vitro would generate a level of complexity which would be very high, would lead to even higher costs than those that we have to incur now. And ultimately, we strategically considered that the best bioreactor to stimulate this cartilage mechanically is the same joint where the cells are implanted. But this requires very careful control of the dose and of the intensity of the loading. And that's why the postoperative rehabilitation regime becomes critical, so that we do expose to mechanical loading these patches, but in a way that is progressive over time and that is not an overloading for a tissue which is anyway not as developed as the native healthy cartilage.
Does the amount of loading during the rehab differ from the loading for other surgical techniques?
We adopted actually a conservative rehabilitation regime that was previously developed following articular chondrocyte implantation. So in this regard, we are in the range of what is typically used. What we try to do distinctly is to try also with certain video instructions to the patient, even when released from hospital and back home, to have rigorously implemented rehabilitation programs.
Okay. It's nice. We've got a good level of interaction here ongoing. There's more questions coming in. And actually, I just see a good friend from Brazil, Tiago, who's also joining us long way online. There's another questions from Poland, I think, from Thomas. What scaffold do you use for cartilage reconstruction and why? And I think you've already answered it.
Yes, so it's a collagen-based material seeded with these nasal chondrocytes.
Yeah. Now, Sazan says hi from Boston. So it's a really international interview today. That's great. Okay, now we talked about your technical approach and about some in vitro data. Now we are excited to hear about the result. Do you already have results on your patient studies?
So all what I'm saying is preliminary, because we have treated as of today, 92 of the 108 individuals that are supposed to be treated in this trial. And we have not reached the ultimate follow-up for many of them. But the main outcome parameter of the study, which is the self-assessed quality of life and state of the joint. So the patient itself judges how does he or she feels as compared to before. And this analysis is done at 1 and 2 years after the treatment with obviously the baseline scoring system. And what we observe in the patients that have reached these milestones is an increase in the overall score and also in the different subcategories of, in average, up to 30 points. And we should recognize here that an increase of about 10 points only is considered to be a successful procedure for cartilage repair. So we are definitely helping patients, making them happy. One of these subcategories is the quality of life or the return to sport. And also for these categories, we have very relevant increases in the self-assessment and in the scores. We are happy to receive videos by patients treated who come back to sport, and some of them go mountain biking, snowboarding. One has run a half marathon, complaining that in the last 500 meters there was a bit of a pain again. But again, we're talking about 20 kilometers. So these are very encouraging results, but only anecdotal, because we have to complete the study to reach the last time point for a follow-up and to be able to derive a statistical analysis of the scores. What is also relevant in terms of outcome is something which is not self-assessed, but which is more an objective measure of the status of the cartilage. And we do this by magnetic resonance imaging, with contrast agent. And this type of analysis captures the biochemical composition of the cartilage over time. And what we in particular can assess using this sophisticated technology is the content of glycosaminoglycans, which is one of the main components of cartilage. And here what we observe is that there is an increase over time of this component of cartilage. So not only we have an immediate filling of the defect with the engineered cartilage, but this new composition, which is a cartilaginous composition, remains and further develops and further increases over time, which to me indicates a correct interface of the graft with the surrounding environment so that it can continue to develop, as we were saying before, in the bioreactor of the joint.
Did you use the same technique to compare the same analysis technique to compare to the standard procedures?
So you know where to hit me.
Sorry, I didn't want to hit you.
No, this is a very important point. We did not have enough capacity to include as an experimental arm in the clinical study a state-of-the-art standard of care control group. So the two groups that were compared in the clinical study that is running are the cartilage grafts generated by the nasal cartilage cells in a stage which is less mature and more mature, to try to verify our hypothesis that the maturation and the quality of the graft can determine the clinical outcome. And then our strategy would be to take the best of these two conditions and to compare this with a state-of-the-art standard of care. That would be done most likely in a phase 3 that still has to start. And one of the reasons why we had to do this was, on the one hand, the lack of resources to run a huge clinical study, but also the fact that there is not a real standard of care currently. So it would be difficult to pick one procedure to compare our treatment with. So we don't know yet, but what we can assess is, again, the biochemical composition of the repair tissue where we introduce our graft in comparison with the surrounding cartilage quality. And typically there is a certain deviation of quality, which even using these most advanced cellular therapies remains of 2 to 3-fold difference in a certain parameter, which indicates that the repair tissue is still not as good as the surrounding cartilage. In our procedure, based on the results that we have till now, we are at 1.3 difference. So we, instead of being 2 to 3-fold different, we only are 30% different. So it's, again, anecdotal and not the correct comparison, but indicates the promise of our method to generate, in the medium term, a good quality cartilage.
It's really promising. And that brings you back to the cells and the difference. What do you think is the mechanism of action? Are they actually doing the job and producing the cartilage, or is it something else?
Yeah. So here is where animal studies can help us, because we can label the cells, we can trace them, we can sacrifice the animal, and we can look whether the cells are still there, if they are contributing to the new cartilage or not. And what we observed in goats and sheep is that they implanted nasal cartilage cells that were labeled prior to being injected in the joint are still persistent in the repair tissue, even in the areas where a good quality cartilage is formed, which does not demonstrate but strongly suggests that they have a direct role in the regeneration of a new cartilage tissue.
There's a question also.
Sorry.
Another question regarding the cells. Fateme asks how many cells you implant into the defect.
Yes. So our patches of cartilage are generated with an initial number of 40. No, sorry, because that's for 2 patches of 20 million cells for a 3 by 4 centimeter patch. This is at the time of loading the cells into the material. What happens when the graft develops until the point in time when it is implanted, we actually do not know. We obviously continue to see many cells living and active, but we don't know exactly how many cells we deliver into the joint.
Now we got a question coming in from Jivko, and he asks, how you decided on the rehab protocol? And he refers to spinal cord injury field, where rigorous rehabilitation is a key for improvement, for patient satisfaction.
As mentioned before, there was an extensive literature search about many possible protocols which are being applied. And we also referred to a Dutch group that is apparently a pioneer or an opinion leader in the field that is applying special treatments of rehabilitation even in the absence of surgical treatment to induce and promote progressive and beneficial, let's say, mechanical loading of cartilage. And this was the basis to generate an algorithm of a treatment that continuously changes, of course, over time, and that is implemented in a video instruction system for the patients. If you're interested in more detailed description of that, please send me an email and I can send you the full protocol and put you in contact with the people who have helped us developing it.
Yeah. If you don't have the email, contact on via our website, and we're also happy to forward it.
Yeah.
Okay, another question from Kiki, who seems to be a real insider, and she asks for the mechanotransduction via shockwaves. Do you have some experience there?
Not really. I'm aware of the fact that there are some external devices that can induce in a non-conventional, not directly compressive modality some stimulus to cells to become more active also in regeneration. This has not been applied in our protocol, but I cannot exclude that this could be an important combination to our protocol to speed up the process of rehabilitation and maturation of the graft. It's an important point, but we have not investigated that.
Thanks. It's an amazing discussion today, but I'm afraid we're running out of time, which is a pity. Maybe out there, the audience, if you would like to continue that discussion in more depths in future, drop some comments, and maybe we can ask Ivan to talk to us again. But before that, there's one more. From Sasan, do we envision using therapeutic approaches or perhaps immunotherapy along with the engineered constructs for some advanced pathological conditions?
So I know Sasan, and he's at Harvard, and I can recognize from his question that he knows what he's talking about. So we know that the immune system has a strong regulatory effect on regeneration. These are thrilling observations and results which are coming out in different areas in the field of regenerative medicine, which have been neglected for so many years. So regulation of the immune system, of the inflammatory events, can have a strong potency on the efficiency of regeneration. And this is another way that we are conceiving to combine with our treatment, so that we not only bring a graft which has the capacity to regenerate cartilage in the joint, but we also condition the environment, especially the immune environment, so that it is enabling and permissive and possibly supportive of regeneration. How to do this? Maybe pharmacologically, by having certain compounds that can reduce inflammation in controlled ways, because we know that inflammation in many instances is also good for regeneration, or by introducing cells which can control the immune cells. And this could be very sophisticated but pivotal breakthrough changes in the field. So
while it's a positive,
but we hope it will come.
While it isn't possible breakthrough, it also increases the level of complexity. And based on these thoughts, I would like to come to like more critical questions, or the limitations of the techniques, so the challenges. What are your thoughts about the manufacturing, commercialization of the technique, cost effectiveness, the regulatory environment?
So in order to generate these grafts in ways which are safe for the patient, there is huge costs. We cannot neglect this. And even if successful, it will be challenging to introduce these costs in a social, let's say, social system that struggles for costs, even for life-threatening diseases. What I'm convinced is that if we manage to demonstrate that this type of treatment can resolve also the degenerative states of cartilage injuries and osteoarthritis, where the costs globally are huge, because if we think of several different treatments, not only the prosthetic implant as an extreme act, but also of the pain of the people, of the absence from work, of the continuous medical visits. So if we can prevent all this, then the costs of production of our grafts will clearly be not only effective, but cost effective for the social system. But we need to demonstrate that this technique is not only valid for focal fresh injuries, but also for more extended degenerative cases. And then I'm sure this will introduce a benefit into the social system.
Yeah. Did you already gain first experience on these indications? What are your next steps?
Yes, so we have in the assumption that there is no good animal model, but that the patient has to be ultimately the environment where we have to study the performance of our grafts. We have treated in 3 cases patients with advanced osteoarthritis. So this is for the experts, Kellgren-Lawrence scale 3 and 4, which is the end stage. So these patients would have had as an alternative only a total joint replacement. And these patients are still walking and slowly returning to sport. So it's even more than before, very anecdotal and very preliminary, but there is promise that there could be an extension of indications also in this space. Discussing with the surgeons, we recognize that the treatment is not ideal, but there may be some refinements to introduce if we enter this type of indications. But I'm hopeful that it will be feasible, and I see a good potential for the future.
That sounds promising, and we look forward to it, to the next presentation on the topic, and hopefully some breakthrough clinically.
Yeah, I'm very happy to have some answers.
So maybe one last question from the audience. until unfortunately we have to finish here. From Chivko, what are the specific measures one needs to take in order to protect the intellectual property on a complex system like this? Wow, that's yeah.
Well, I can just, in the sake of time, be very short here by saying that there is no possible future of these technologies if there is no commercial partner. And there is no commercial partner if there is not a way to protect the intellectual knowledge on these treatments. And currently the use of nasal chondrocytes, the use of these scaffolds, the techniques that we use to engineer grafts are common domain. So there is nothing to be protected. And our strategy, where we see a potential for intellectual protection and so for attractive interest to company and private groups, is in the development of bioreactor systems, so controlled devices that automatically, in standardized ways, produce the graft. So there is different pieces of technology in the process that need to be introduced that can be patent protected. And we have one such patent already for one particular system. And with that package, I'm convinced that an industry group might find the procedure attractive also for commercial exploitation.
Thanks a lot. Maybe if I can take the freedom to ask one last question from my side, because I'm just too curious. Imagine being a patient. How do patients react when you offer them to extract cartilage from the nose to fix the knee?
Yeah. So far, the patients have been extremely open to this procedure. Also because the surgeons are well trained to explain that it's less invasive to be biopsied in the nasal septum. This is a procedure under local anesthetics. In 30 minutes, it's over. The patient can go home, as opposed to entering the knee and to the articular space to get a biopsy of the cartilage from the knee, which is definitely more uncomfortable and may have long-term negative consequences. Okay. So if a patient receives this type of information, we have not experienced actually any retraction because of that.
Okay, that's good to know. There's a comment by Kiki, and I think that's a perfect last words, and she says, great talk and study. Really appreciate the hard work. Looking forward to the publishing paper and more promising results. And I can only confirm this. And thank you very much, Ivan, for that exciting and stimulating talk.
Thank you, Kai. Thanks to everybody who followed and participated actively in the interview. It was a pleasure.
And maybe out there, audience, if you would like to continue, if you've got more questions, drop us some messages and we'll try to facilitate it. Okay.
Very good.
Bye-bye.
Thank you. Bye-bye, everyone.